we first have to make a blood smear, and that is made on a glass slide, and for making a blood smear we require another glass slide. So a drop of blood, 1 or 2 drops of blood are to be taken on a glass slide just near one end, that means one end which is the breadth, not along the length. You will consider the breadth, so this breadth end, so just near, just in front of one end you take one blood drop, keep it horizontally on a surface, and then with another slide, just you tilt it like this, maybe roughly at an angle of 45 degrees, put it in front of the blood drop. Okay, you have to put it in front of the blood drop, then slowly pull the slide backwards till the edge of the slide, this edge of the slide comes over the blood drop. Then what will happen, that blood drop will spread along the blood, one drop or 2 drops, whatever it is, that blood will spread along this edge of the slide. Then with one single stroke, say suppose it is a horizontal surface, so for showing it I am holding it up, so it will be on a horizontal surface. With one single stroke you draw the smear. This is what is called the blood smear. With one single stroke, that is very important. Otherwise, if you just fumble like this, then there will be distortions in the smear. This is called a blood smear on a blood film. So it will be difficult to stain and difficult to identify cells. Okay, difficult to stain, because the next step is staining, which those who are new will just show you. So over the blood drop, along the whole length of the smear, you have to put stain. What is the stain? The stain which we use here is called the Leishman stain. It has eosin, methyl alcohol as the staining components. So we call it eosin and methylene blue, and methyl alcohol is the base of the, that means of the solution. Eosin and methylene blue, we call it eosinated methylene blue. Eosinated methylene blue in acetone-free methyl alcohol. Why it has to be acetone-free? Because acetone cremates the cells. Okay, so acetone methyl alcohol has to be acetone-free, and it will be eosin and methylene blue, which has the staining components. Eosin stains cytoplasm of RBCs and granules of eosinophils. Nuclei of the WBCs, granules of other cells, wherever there will be granules, except eosinophils, granules are present in basophils and neutrophils. Lymphocytes and monocytes have no granules, so they will be those granules, everything except eosin. So remember like this, except granules of eosinophils and cytoplasm of RBCs, whatever is being stained by methylene blue, and you will find the deep purple color. RBCs you will find as pinkish round structures under the microscope. Those are for identification of cells. But before that, the procedure of staining is important. Put the stain. For putting the stain, those who are new have to count the number of drops, how many drops of Leishman stain are covering the blood. Then after waiting for 2 minutes, you have to add buffered water. If it is distilled water, here buffered water is available. Buffered water, equal number of drops have that of the Leishman stain. So counting of the drops is important. If these quantifications are without following these quantifications properly, the cell will either be overstained or understained. So again, it will be difficult to look for cells under the microscope. Okay, so it will not be perfect on the first day, second day. So, but you have to follow the procedure. Try to follow the procedure correctly, especially those who have joined newly. Okay, before that, how will you get blood? You have to prick your own finger with the help of we collect capillary blood. Capillary blood is collected by pricking the finger. In cases of children, it can be the heel, it can be the earlobe, but the common practice is to use the middle finger or the ring finger, and near the tip of the finger, that means the distal most digit of the finger, middle finger or ring finger, and somewhat near the ulnar border. Ulnar border means again the medial border of the finger. Okay, so this area, these are chosen. So this should be a single deep prick, so that blood oozes out. Every word is important. So that blood, why are you laughing? So what is there to laugh? So just these things will happen. So you don't get an expert in one day. So there is no reason to laugh over this. So the very important point is single dip prick so that blood comes out freely. The word is free flowing blood. Why free flowing blood is important? Because if you don't get free flowing blood, obviously how will you collect the blood? So blood has to be. So if say just blood is not free flowing, just even not a drop, not a single drop is coming out, then the common thing which comes to layman, even we try to squeeze out blood, that will require blood, so now we try to squeeze it out. Now what happens? Blood get mixed with, actually blood does not come out, what comes out more is tissue fluid. If blood gets mixed with tissue fluid, more of it is tissue fluid, blood cells are not contained in tissue fluid. So again it is difficult to, that means not possible to do the experiment. You can't, what will you count if there are no cells? So blood has to be free flowing, and one obvious thing is needles one needle cannot be passed on to another individual. It can be used more than one time, but it is recommended to use one time only, but it can be used more than one time, but for the same individual, obviously not for other individuals. So the same needle, and you have to be very protective about how to. So just take the drop of blood, you have to keep the needle by the side, you have to again put the cap on the needle and then put it. Don't throw away the needle or keep the needle somewhere where it may hurt someone else. And for using the microscope, there are some, we have our first class was on the use of the microscope. However, you must have learnt it in your school days, but for using the microscope, there are some precautions to be followed. For those who are new, we will discuss this. You just go to the tables, we will discuss this, how to handle the microscope and what are the parts of the microscope. I will just revise with you. Okay. Now, those who are new, please make groups. When we will settle into groups, then please join some of them who are attending the classes from earlier. So you passed your class 12 exams just this year, last year, and you were preparing for NEET. So this is the microscope. These lenses are called the objective lenses. I think you know, but just for recapitulation, those objective lenses magnify the object by there is these magnifications are written. So this one is magnifying whatever object will be there, it will magnify it by 10 times. It is written over here, 10x. Okay, the x signifies the magnification. That means how many times it is magnifying. One is there which is magnifying 40 to 45 times. Another is doing it 100 times. For counting the cells, we may use the for counting this topic, differential leukocyte count, we have to use the 40 first for identification, 45, and then we should use one oil, that is for matching the refractive index, oil and the 100 times magnification. And total magnification, this is one lens and this is one lens. This is called the eyepiece lens. This is also magnifying the object 10 times. So total magnification would be multiplying the whatever lens we are using, the objective lens and the magnification of the eyepiece lens. These two will have to be multiplied. Usually for counting cells is 100 for the objective lens, the 10x eyepiece lens. So total magnification is 1000 times.